So without any further delay, Dr. Mason.
This is wonderful because I have to say, after 25 years, it's long overdue that I should visit my favoritely named medical school. Thank you. It's a delight to be here on the other side of Chicago. And so what I'm going to tell you about today are studies that I started. I didn't actually start them. A student started them in 2007. So involved Enemy Bartal, who's right here. Was a graduate student at the time, was John DeCidi, and she'd been looking at empathy in humans, and she'd had some success at discovering some interesting findings, but she missed rats and mice, and she basically thought, I actually am not so interested in this. I want to look for a biological basis of helping. Is there a biological basis for helping? Is there helping in a non-human animal? And so at the time, we already knew that animals could communicate affect between each other. And we knew that. And what Inbal wanted to do was to take that one step further, a big step, a leap further, which is if they can if they can catch the affect of another, can they use that to motivate their own behavior? So what had been shown was that if one rat is frozen, another rat can freeze just seeing this frozen rat. But that doesn't, that's an internal experience. It doesn't really do much. What you really want to do is sort of take that motivation out for a drive and try and get to the speed limit. And so the way we conceived of our challenge was to build a task, a helping task that was rat-sized, okay? And so this is basically what we came up with. We use an arena that's about yay big, and in the center of the arena, a place where rats naturally don't want to be. The center is scary. That's where they get picked off by the hawk. They'd rather be at the edges. So at the center of the arena, we put this clear plexiglass tube, which we call a restrainer. It has one door on it, and that door can only be opened from the outside. So we have a trapped rat inside the restrainer, and we have a free rat outside who can, should he decide to do this, open the door. So what we found, so the question is, will they actually not just catch the other rat's distress, but actually do something with it? And the do something, it seems easy to you. Go open the door. Well, first of all, the rat doesn't know what's a door. And second of all, it takes a while to figure out how to open it. So even if we say put chocolate in there, it still takes them 3 days, 4 days to figure out how to open the door. Okay? So, and I would be talking to you about this if they didn't open the door. They do. So the way we fashion this experiment is that they have these daily sessions for 40 minutes, and we repeat them over 12 days. Now, what are we looking for? We're not, if they open the door, it's kind of a so what. That it's probably by mistake. First time the rat opens the door, it's probably by mistake. So what we're looking for is them opening the door again, and then again, and then again. We want to see consistency, and we want to see that they are faster and faster at it. And indeed, that's exactly what happens. So these are data from our very first publication back in 2011. And what you see in blue is the percent of rats that open on the left. And then across the testing days, the percent grows. Only if the restrainer contains a trapped rat. If the restrainer is empty, or it contains an IKEA toy rat, they don't open. So they need to be motivated by this trapped rat. And not only do they do it more and more often, more of the rats do it, and more of the rats do it on each day, but the time that it takes them to do it goes down across the days of testing. So by the 12th day, what they do is they get in there, they run around for a few tens of seconds. Oh, same gig as yesterday, now I'm going to open the door. Okay? And they open the door within a minute or two. So what I'm going to show you now are some videos. And this just shows you the first one is the first day of testing. It's sped up by a lot. And the only point here really being that what you can see is that the rat goes in. He wants to be on the side because that's a safe place, but he keeps on going in to check on this poor trap guy. You know, what's going on there. And that was remarkable. When we saw that, we kind of knew we had the whole thing. Now this is real time, but it's 25 minutes into the testing session. And what you see is he's going to open the door. The door is going to make a sound, which you may be able to hear from my computer. And when it makes a sound, they're both going to freeze because they're surprised by the sound. He's never opened the door before. So that's not a glitch in the video, that's them freezing. And now what you see is that the guy who opened the door follows the trap rat. It is always in that direction. It is always the free guy that follows and jumps on and licks the trap guy rather than the other way around. We can talk about what that may or may not mean. Now this is the last opening. It's real time. You can see that he's just been put in. He runs around. Same gig as yesterday. We barely have time to put the lid on. And then he's going to come over here, open the door. And what you notice is there's no freezing. So this is now day 12. He first opened on day 6. And so that noise is now the expected consequence of an intended action. He knows that's what's going to happen. Okay? So that's what the behavior looks like. Now, once please do ask questions throughout.
Maybe I missed it, but are they both male?
They're both male. We've done this. Those of you who know me will understand how painful this is for me to say. We've done this with females, 2 females, but much, much less. I have no excuse. I could tell you some historical reasons, but there is no excuse. And almost all of our work is with males. Okay, so once we had this, you know, we don't know what's going on. The simplest explanation, the most obvious explanation for this is that the free guy is kind of lonely and says, oh, I want to play with the trap guy. Let me get him out so I can play with him. That's a very reasonable explanation. So we wanted to see whether the free rat, in fact, opened the door in order to play with the trap rat. And we designed this experiment where we actually put 2 arenas side by side, and between them there was a barrier that had a hutch in it where the restrainer went up to. So now when the free rat opens the door, the trap rat is released into a separate arena. The free rat cannot play with the released trap rat. And we did this experiment for a long time. We did it in 2 groups. One where they started with an empty restrainer. So we expected them that they would eventually extinguish, they would stop opening, and they did. And then when we put back in a trap rat, they started opening again. And one where we started with the trap rat, they continued to open for the trap rat, and then when we went over to an empty restrainer, they stopped. So this was this has convinced us that, in fact, the free rat does not open for the trap rat in order to have direct social access, in order to play with him. in a very happy event, any of you who publish a new paradigm, you'll know that the first time somebody else actually publishes using your paradigm, it's a celebratory event. And that did occur in 2015 with Nobuo Sato. And he adapted our paradigm in a really interesting way. And he showed the same thing, I think, in a slightly different and in fact in a slightly more compelling way. So I'm just going to show you what he did. His task was that this is the free rat. Here's a pool of water. There's a rat that is called the soaked rat, who's in this pool of water. And in that situation, this rat will open this door to let the soaked rat out. Now, if the rat is over here, which is the same size, but has no water in it, he never opens the door. Okay? So he's not opening the door to play with the guy. He's opening the door only when there is distress on the part of the rat in the other arena. Okay? Yeah?
Does having been trapped?
No. Okay, so we never did that, but Nobuo did, and having been trapped speeds up your learning. Yeah. Oh.
Oh, no. Not moving?
No, I'm it's trying to tell me I'm out of memory. Okay, so we were convinced that it was not, they just want to play with each other. And because they don't open for an empty restrainer, that takes away sort of what I call the Mount Everest explanation, because it's there. They're not doing it because it's there. They have really no interest in the empty restrainer. And building upon the evidence that this is a robust, complex, motivated behavior is the fact that they won't open for everybody. So this slide summarizes a large set of experiments that demonstrate that they will open for rats of a type that they are familiar with, no matter whether they are familiar or not with the individual. So in other words, an albino rat will open for another albino rat, whether they've met or not, but will not open for a black cape rat if they've never met a black cape rat. They will open for a black cape rat if they have lived with one black cape rat for 2 weeks. Lived with one black cape rat for 2 weeks, they'll open for any black cape rat. So those findings there told us, back when we were doing this, that environment was very influential on who a rat would open for. And the obvious question was, does genetics play a role? And so what we did was we did an experiment where we transferred our pups, albino pups, into a black cape rat litter on the first day of life, on P0, and they grew up, these albino pups grew up in a black cape rat environment with black cape rat mom and litter mates, and then with a black cape rat litter mate cage mate. When they were adults, we tested them, and what we found is, of course, they opened for black cape rats. They've been living with them for all their lives, but the real question that we wanted to know is, would they open for albinos? And the answer is no, they will not. So genetics is insufficient to tell them that they are, in fact, to breed familiarity and to breed the willingness to help that type of rat. So that's a pretty, that was very exciting to us. But I'm not going to talk about it. I just told you all about it. Okay, so so what I am going to tell you about is our work trying to figure out what's the motivation for the rat to open the door. And recently I decided that I sort of wanted to explore this in a bigger picture. I wanted to see what's the space in which we have some variation. What's the dynamic range? So I wanted to know what's the floor and what's the ceiling for this effect. So for the floor, we just redid the experiment where it's an empty restrainer. And for the ceiling, what we did was we took rats, we put them in the restrainer, and we turned around the door so it could only be open from the inside. So now they're not opening for another, they are opening for the self. That's the, that's the ceiling. Now it's a slightly easier task, so it muddies up. The ceiling's is a little higher than it should be, but I think you'll see the point. Okay, so here are the results. So in blue, what you see is an empty restrainer, and the median latency is 40 minutes, which is the length of the session. In other words, they never open. In fact, there were 8 rats tested, and only 1 rat opened on one day, the 12th day. And we're interested to say, oh, well, maybe that rat, maybe there are rats that get a kick out of opening an empty restrainer. So we tested them one more day. We wanted to know whether this was a joyful experience for the rat. And in fact, on the next day, he didn't open and no other rats opened. So this was the only opening in 13 days, and it was an isolated single event. On the other hand, when the rat is releasing himself, you can see that most of them release, a majority of them release already on the first day, and they release at very short latency. We're putting them in, and they're opening the door right away. It's under a minute. Okay, so now where in this space does helping another rat fall? And so this is a group of 24 rats that just went through the basic paradigm. 1 rat opening for his cage mate, and what you can see in red is that it's much closer to the ceiling than it is to the floor. Now, when you take different control, if you do behavior, you'll see that rats have minds of their own, so to speak, and they're not going to home to the average all the time. So here's a separate group in these red symbols with a black outline, and what you can see is they still occupy the same general space, but they're not overlapping, they're not identical. And stuff happens. So for example, here, a bunch of them opened on the first day, but no one opened on the second day. Why? Well, very possibly because there was a big commotion in the hallway that day. So there's a very sensitive behavior, anything can happen, and the gemisch here, or the gestalt of this, is that this middle space is actual helping. It's far, far away from what you would do for an empty restrainer, and it's not so far away from what you would do for yourself. Okay. Now, another criticism of our work, besides our own self-criticism, that could be that they just want to play with another. And actually, I think this happened. I was talking to an Irish public radio show way back in 2011, and they this radio person was just sort of saying, well, what else do they have to do? And it always stuck with me. And other people said it in slightly more biological terms, but seriously, what do they have to do? This is an impoverished environment. It's an arena, and there's the thing. So you got nothing else to do, you might as well open the door. So what we did to look at that was to think about what else is there to do. Well, we gave them some other things to do, and we call this the toys experiment. So there's a restrainer with a trap rat in the middle. There's an open restrainer here. Now, you should be aware that an open restrainer and a closed restrainer are very different beasts. That you cannot make them be happy about being in a closed restrainer. We tried a lot. We gave them all sorts of goodies that only enclosed restrainer, but no, they will, once that door closes, they hate it. But they like the open tube. They'll go there voluntarily and they'll stay there. We also put in a ball of tape. We put in this EnviroDry, which is a nesting material that they like to play around with, and a door, because oftentimes we see them playing with the door after the door is off. Okay. And so what do we see? Well, we see that, in fact, yes, opening takes a hit. It's less than it would be normally. Essentially, it starts later and fewer animals start to do it. But those that do actually do help. And so it's a delay. It takes an impact, but this is definitely still a motivated helping behavior on a good portion of the rats, on the part of a good portion of the rats. Okay. So now that we believe that they're actually motivated to do this, and they're motivated by the trapped rat, presence of the trapped rat, we wanted to know the simplest explanation, the explanation that we favored, in fact, is that they are moved by an affective experience to help the trapped rat. This is not, it's not a cognitive story. Gee whiz, I would not want to be in that tube. This rat is in distress. I catch that distress, and feeling that distress is a motivator for my own action. And so if that's true, what we should see is that once they actually open, they receive something that's good, some goodie, and that then carries over to the next day. And that's basically what you see. So here I'm showing you 16 rats that are in the basic paradigm. And these are the 12, each column is 1 day, so there's 12 days of testing. Each row is a different rat. And if they open once in a row, that's yellow. And if they open 12 times in a row, which none of these do, it would be red. And so you can see color-coded. What they do is they open once, and then there's pretty much a 90%, 80 to 90% chance that they'll open again. If they open twice in a row, it goes up close to 100%, and by the time that they've opened 3 times in a row, it's 100%. So this is a very reinforcing behavior. The way that we interpret this is that it is a switch. It's a switch of reinforcement. For those rats that are going to open again on the next day, they got something really good on this first day, something that made them crave doing that again. And just another way to show the same switch, if we graph the latency of their opening on the 1st, 6th, and 12th day, you see on the first day, almost all the rats are up at cutoff at 40 minutes. A few of them open. By the 6th day, more of them have made the transition down to a very quick opening. And by the 12th day, they're either not opening or they're opening at very short latency. So there's not this gradation, and no one lives here, no one persists here in the middle. You make a switch from not opening to opening. Okay, what motivates that? Is it affective in nature? To ask this question, what we did was we basically just injected the free rat with a chill pill, which is midazolam. Okay, so this is the benzodiazepine. It's an anxiolytic, and it's going to make them a little bit affectively flattened. And so we injected the free rat, and we tested the free rat, injected with a low dose, a high dose saline with a trapped rat. But benzodiazepines also have a sedating effect, And so we wanted to make sure that any result we saw, what was the effect of the sedation. So to test that, we also tested rats that were injected with the same drugs on chocolate. Okay? So if it's sedating, they won't go get the chocolate or the trapped rat. And what we found was really quite remarkable. I'm going to show it to you in two ways. This is sort of the drier way of showing it. If there's a trap cage made in their strainer and they get saline, they still open. The proportion of rats that become openers still climbs across the days. If they get a low dose of midazolam, it climbs across the days a little bit less. And if they get a high dose of midazolam, they do not open. Okay? On the other hand, if they get a low dose or a high dose of midazolam, they open really, really well for chocolate. Okay? So they are not sedated into a stupor, and that is not the explanation for why they're not helping. They are not helping because they are not motivated to do so. They don't catch the distress. They don't catch the distress of the trapped rat. So here's just a different way of looking at it. On the top, in the same way that I showed you before, each row is a rat, each column is a day. This top group of graphs were rats tested with a trapped rat, and the bottom are rats tested with chocolate. And you can see if they got saline, they make the switch into helping. If they get saline, only one of them makes the switch into getting chocolate. Think about that. Okay, and then if they get, on the other hand, if they get the high dose of midazolam, no one really becomes a helper, but they still go for the chocolate. Okay, so which is more reinforcing, chocolate or helping a trapped rat? And the answer is definitely a trapped rat, helping a trapped rat. Yeah?
What if the trapped rat is sedated?
We're going to get there.
Must have been a good question.
It is a good question. Okay, before we get there, I want to take one sidelight. So what we think is that the trapped rat is distressed. The free rat catches that distress, and that is motivating for the free rat to then go and relieve the shared distress of the trapped rat's predicament. But there are always about 25% of the rats that don't help. And one can wonder, what's up with that? Are they psychopaths? And we were looking for psychopaths, and we might have seen a couple, but not enough to study. The other alternative is that they are actually overwhelmed by too much caring. And that is, in fact, the answer that would be favored by human research, that people that are extremely affectively affected by other people's predicaments and distress are overwhelmed and cannot make other-oriented actions. And in fact, that's what we saw. So what we did was we exposed the rats to a trapped rat, and we measured their corticosterone response. The rats with the highest corticosterone response were the rats that then, when we then tested them on the next 12 days, proved to become non-openers. So the rats with the highest corticosterone response, the rats most affected by seeing a trapped rat, are the rats that will become non-openers. Okay? So it's very, this is, in the human literature, this is called downregulation. You really need to downregulate your own emotions, your own affect in order to help others. Yeah?
What do they do instead?
They stay on the side. They're just plastered to the edges. They don't go into the center. Yeah.
But could it be that they are communicating with ultrasound like mice do?
So the question is, are they communicating with ultrasound? And the answer is that we don't know the sensory signal that is used. We don't think it's sound because we did measure sound in the first experiment, and it's only 13% of the sessions that have any 20 hertz alarm calls. So we don't think that's the major thing, but in fact, we don't know. Okay, so as I said, working hypothesis is the free rats catch the distress of the trap rats and help in order to alleviate that distress. Well, if that's true, then what happens if you block the trap rat's distress? Okay, so now what we're going to do is we're going to say, does the trap rat have to give off affective signals? We're going to interrupt his affective signals by giving. We're doing the same experiment, but now we're injecting the trap rat with benzodiazepine, midazolam. And what we saw was that it takes a hit, helping takes a hit, but they still get helped, even if they are injected with midazolam. So what you see is in black, these are the rats that are injected with saline. So a bunch of them get help. They get help by day, say, 6, and at short latency. And if you give low dose midazolam, they get helped a little bit later. And if you give high dose midazolam, fewer of them help, and as a result, the median latency of helping is much higher. So helping takes a hit, but this is not what we saw when we gave midazolam to the free rat. And so the first idea that we had was, okay, well, we think we're chilling them out, but maybe we're not. Let's ask them. Let's see how chilled they are. So we gave them the midazolam and we put them into the restrainer with the door reversed. If you're chilled, you can hang out there. You're not going to get yourself out. But if you don't like it, you'll get yourself out. And so what they told us is that, the midazolam did an okay job, but it did not block their distress. So what you see is uninjected rats are down here. Saline rats are even more, or get themselves out at even shorter latency and faster. And then when we give high midazolam, they don't get themselves out as quickly, and not as many of them, but they still are getting themselves out. Okay, so we did not obliterate distress. So that's one answer, that's one potential answer why they're still getting helped when the trap rat receives midazolam. So then we say, well, what happens if we completely obliterate the rat's affect? So we did this in 2 ways. One was an enormous dose of midazolam, a sedating dose, a really sedative blotto dose, and the other one was with anesthesia. And we combined those 2 groups, and what you see, to our great surprise, is they get helped. So this is what happens. And this happened on the very first day. Peggy, we put the anesthetized rats in there, and the guy makes pretty much a beeline, opens the door, and then he drags the trap rat out. All right? And then he just, he's a little fixated on the trap rat who's motionless there somewhere in the arena. So what we realized at that point was that there were many stimuli, there were many things that could be responsible for this. One is that what we know that we obliterated is the trap rat's distress. So the trap rat, this is off the table, but there's 2 things that are still on the table. One is the situation. There's a tube, and you're in the tube, and poor you, I wouldn't want to be in the tube. You must not want to be in the tube. That's pretty cognitive. Maybe that's driving it. And the other one is the rat looks weird, and we call this the dead or alive. Let me explain that a little bit more. We imagined ourselves in University of Chicago. We have a nice quad. We imagined ourselves walking through the quads and seeing somebody sleeping. As we pass this person sleeping on the quads in the summertime, we pass on by. We don't think twice of it. We go on our merry way. We see somebody now. Let's imagine that we see somebody who is passed out, not doing well. Now you look at that person, you realize that person's not doing well. I should do something. I will call somebody. I will see whether they are all right. I will try to help them. And what we hypothesized was the rat was doing the same thing. He was having the same experience. Not because of the situation. He didn't realize that the tube is an unpleasant place to be. But he did realize that the rat looked weird. He looked off. And for whatever reason, that doesn't sit well with the rat. So what we did to test this idea was we placed these anesthetized or highly sedated rats in the middle of an arena, but not in a restrainer. So we've taken the situational issue out, and all that we have is this off rat. Here's this rat that looks weird, basically because it has no tone, it has no posture. And what do we see? We see that the rats push around this. So the anesthetized rat is the one with the star, and this is the awake behaving rat. And he looks at it, he climbs on top, he bites him to try and get him to do something. Well, I'm saying that, I don't know. But he does bite him. And he nudges them, and they nudge them so much, they get them out of the center, they push them over to the edge, and then they push them into a corner. When do they do this? They do this when the rat is off. They do this only when they're tested. These behaviors really only happen, pushing out of the center, the tail biting, these only happen when they're encountering an anesthetized rat. So we would advance, and I'm going to say this, knowing that I'm going to make a lot of people unhappy, but I would advance that this qualifies as a cognitive form of empathy. And I say that because in these animals, there are no affective clues available. The only clues that are available to them are this rat looks off. That's a cognitive realization. It does not depend on affect. Okay. So, great. So yeah, we haven't published this yet. When we publish it, I anticipate more hate mail. Yeah.
Do they cannibalize rats that pass out or die?
We have never seen them cannibalize them.
He's pushing it into a snack corner for later.
We've never seen them cannibalize them. Yeah.
If the rat is anesthetized throughout, will the other rat actually learn to open the door?
They do open, yeah, they open the door.
Oh,
Yeah,
that's
this. The red is, these are anesthetized rats. They're opening the door for anesthetized rats.
but they're not trained first with the weight rats.
No, they're not trained with, no. First day, out rat. Okay, so I do have 15 minutes. I guess I've talked fast. I apologize for that. And I'm going to tell you one last story. And this is a story that was initiated by a psychology student at University of Chicago. Well, actually, he's a neuroscience major now, because we now have a neuroscience major, but he is very interested in psychology. And he came to me a couple summers ago and said, let's study the bystander effect in rats. Now, I had thought about this for a long, long time, and I had concluded that I didn't know how to do it. But John Havlick, a really talented undergraduate, really pushed me, and we actually figured out a way that would be informative. And then the next summer, he was joined by Mara Clement, a very talented medical student. And they've done this, what I'm going to tell you is the sum total we've done on this, but it's pretty exciting work to us. So let me start by telling you what the bystander effect is. This was an effect that was described best and most completely by Big Latame and John Darley. They were motivated by the story published in the New York Times about Kitty Genovese that story is apocryphal. It is not, the story that was published in the New York Times is not actually what happened, but nonetheless, they were motivated by that apocryphal story, and they wanted to see how the presence of others affected an individual's willingness to help. And they investigated this in a wide variety of conditions, but I've chosen one very easy to understand one to share with you. And that is, a person comes into a room, they're given a questionnaire. The questionnaire is not part of the, important to the experiment, but it is important that they think that they're there to fill out this questionnaire about how they're adjusting to college. And so they fill out this, but somewhat time during the session, smoke comes in through the return in the room. And so smoke is coming into the room. And if they are alone, 75% of them get up, open the door, go out, and say to somebody, smoke's coming into the room. Now, another group of subjects is tested, same situation, except that there are two people there, and both of those people are Confederates, but they are told not to do anything. And in the presence of these two others, only 13% of the subjects actually leave the room, okay? So the presence or absence of these two individuals has changed the behavior of a subject. That is, in essence, a social behavior. That's a social modulation of behavior. And so what we decided to do was just to add rats. So what we did was we put 3 free rats on the outside and 1 trapped rat. And that's what John did on the first summer, and this is what he found. It was, so the control is the regular paradigm where there's one free rat, 1 trapped rat, and then this black is where there's 3 free rats. And what you can see is that already by day 6, they're all opening. And so we cut off the experiment at that time. I'm kind of glad we did. I'm kind of not glad we did. But and I won't go into this variety. We can talk about that if it comes up in questions. But the point being, at a first glance, if you take the bystander effect to be that individuals are less likely to help in the presence of others, that does not occur. That did not occur here. Okay. But as I said, in these psych experiments, most of them are done not with just naive subjects, a group of naive subjects. They're done with Confederates, people who are part of the experimental team and who are instructed to behave a certain way. So we did the same thing. We introduced Confederates. Had we do that, we gave free rats midazolam, and we know they won't help if they have midazolam. Okay, so we midazolam them into being Confederates. And using either midazolam injected rats or not, naive rats, we could make a scale of individuals in the arena from -2, meaning there are 2 Confederates in addition to a subject, or 1 Confederate and a subject, or 0 is just the regular paradigm, 1 helper, one free rat, 1 trapped rat, and then +1 is an additional naive rat, and +2 is 2 additional naive rats. So, and you can see here, this is with 2 Confederates. They're both chilling out over here on the side. They don't go into the middle, whereas if it's 3 rats that are all naive, they all go into the side, into the middle, and have a lot of restrainer-oriented behavior. So what we see is that every metric of opening is affected by who's in the arena. So for example, how many of them opened on day 6? Well, if there's 2 extra naive rats, it's 7 out of 8, whereas if there's 2 Confederates, it's 3 out of 8. If, how many openings per arena? We did through the 12 days, well, we only did it for half the days here, so that would be approximately 11. This is 10, and then when you put in Confederates, there's only 4.4 openings per group. And so, even though there are more rats available to open, and so here is, again, from the scale of -2 being 2 Confederates to +2 being 2 naive rats extra, the number of openings per rat on days 1 through 6, and you can see it goes up. And the reinforcement, the first day on which they then follow an opening with another opening, that gets earlier and earlier as you add active rats. Our interpretation of this is that the bystander effect is a very biased term, or the way that we define it's commonly defined, say, in a psych textbook. It's a very biased term to first of all talk only about helping, and second of all to only talk about a decrease in helping. What we think is actually going on, and which has a lot of resonance with Connie and Darley's findings, is that the rats at every moment they're incorporating not only their own sensory, their direct sensory input, but their perception of others' sensory input. So why would you be in a group and ignore what you think others are thinking? You know, if I go into a place and everyone's avoiding some part of the room, I think, well, okay, for some reason, I guess I should avoid that part of the room. If I go to a food buffet and I see that no one's eating a particular food, I think, oh, well, there's something wrong with that. I'm not going to eat it either. Although it might be under normal circumstances, one of my favorite things to eat. So we are influenced, and it is a good thing. Obviously, one can imagine it would be evolutionarily advantageous to incorporate others' perceptions with one's own perceptions. And that's what we think is happening. And indeed, if you ask Vivian Latani and Darley's subjects what was happening when they were alone in the room, they thought, okay, there's smoke in the room, there's a problem. But when there were Confederates in the room, they made up all sorts of stuff. They said, oh, well, I thought the smoke was just there to get us to tell the truth. I thought it was there and it was a sign that we were doing the questionnaire correctly. I mean, they just made up all sorts of crap stuff. Excuse me. And so if our interpretation is correct, then there are a couple of corollaries to that, which are things that we're interested in, which is that it shouldn't be confined to helping. So in other words, if I had a bunch of rats if I have a rat that likes chocolate, and then I bring in a couple of rats that have a conditioned taste aversion to chocolate, that one that likes chocolate, if the conditioned taste aversion rats are around, will probably not go to the chocolate quite as readily. And conversely, you can imagine that if a rat doesn't like, say, quinine, and is in a situation with rats that love quinine, that run to get the quinine, that rat may, in fact, try the quinine. And this has some interesting applications in husbandry, because many zoos and aquaria are interested in switching their wild-caught animals from a wild-caught diet to a non-preferred diet. And the idea being that if you see that switch with other animals who have already made the switch, you can actually make animals switch faster. And so that's something that we're exploring with the Shedd Aquarium. And with that, I will end and say thank you for your attention. Here are the people, and I'm happy to take any questions.
TV show, what did you do?
I do not.
If they do these type of experiments, I didn't need no. And all of them, too, they've been doing this for several years.
I mean, this is very Candid Camera.
Yeah.
So I don't know about that show, but Candid Camera's from the 50s. And one of my favorite Candid Cameras is you're in an elevator, and all of a sudden, one person starts, instead of being headed towards the opening, heads to the side. Slowly, everyone follows them. And you go this way. And slowly they'll follow me. You know, I mean, you can just make people do it, whatever, if you do it with enough zest and assurance.
So is that behavior, motivation for this behavior is establishing social status? The alpha male helps the beta or
So this is a really
male.
good question. The problem is that in mice, it would be easy to answer, right? Because we can measure our hierarchy in mice pretty easily using a tube or something. But in rats that are housed in pairs, there is not an easy, obvious hierarchy. So we don't know. I mean, if I could house my rats in 5s or 6s, I might be able to establish a hierarchy and answer that question, but I haven't been able to do that.
Because you.
But what I can tell you, let me just say one more thing, which is that we've looked at every once in a while, like with strangers, for example, the weights aren't right. And what I can tell you is it doesn't matter whether the trap rat weighs significantly less or significantly more. They open either way. So that we have a meta-analysis that's clear that weight differences make no difference. You know, weight is not hierarchy, but.
Because the corticosterone study shows the correlation with elevated force level and lack of empathy, which usually comes together with lower status.
Yeah, I know that it's a reasonable hypothesis. We just haven't been able to do it because we don't have
I
a
know the
hierarchy.
association with another study of autism in birds, Egyptian finches, in which the alpha male is always on the top of the tree, exposed to predators, and of course he gets more mates. So it's a part of their sexual behavior, and again, the alpha male is on the top, exposing himself. So that's why I made the association.
Yeah, no, I think
Maybe
it's a really interesting
start to
question, and we've thought about it. We just haven't. We got nothing so far. Yeah.
Any thought to see whether the behavior of non-helpers could be altered by running them with responders?
We haven't thought of that. We have thought a lot about trying to give the non-helpers midazolam to bring them. What we think is that, you know, if you have a sort of a core stress reactivity scale versus helping, it's going to be a bell curve. Too little, too much, both problem. You want to be right there in the sweet spot of the Goldilocks middle. The problem is that we don't have any fast way of telling, oh, you're going to become a non-helper, you're going to become a helper. So 25% of them don't become helpers, but we don't know that going in. We don't know which ones it's going to be going in. So we haven't.
I mean, from the data you presented, it looked like you tested core.
Yeah, but that is not a quick predictor. That's not something where we can say, oh, let's test this today, and tomorrow we'll split them according to these groups. Yeah.
So the 3 rats somehow perceive the other rat looks off. What would happen if the cage were black instead of plexiglass?
Oh, the cage is black. It's black plexiglass. You know, I don't know if you can tell. I mean, this is the flash is making it gray, but it's black.
So what's the look?
Oh, the look of the rat. The look of the rat.
Are you
I mean-
asking about making the restrainer flat?
No, I'm saying the restraining cage flat.
Oh, the restrainer cage, no, we haven't done that. I mean, again, what's the sensory cue that tells them that there's a problem with the trapped rat? People are gonna point to ultrasonic vocalizations and olfaction as the top two, but I would advance that vision probably plays a role, and the one that, if you watch them, the one that seems to play a huge role is taction. So there are a lot of holes in the restrainer, and they put their vibrissae out, and they do. So I think they're really having communication vibrissally.
You have a question from Facebook from Norway. And Trina wants to know, can you make available links to the papers or articles about this?
Yeah, it's all on my blog.
On your blog?
Yes.
Blog thebrainissocool.com.
Indeed.
Thebrainissocool.com.
Yeah.
So you can't a priori tell who's going to be a helper
Right.
or
Right.
indifferent. Have you considered looking at the progeny of those rats that are known helpers or known indifferents?
We have, and we actually are about to do this experiment where we're going to see, we're gonna do this in a totally walk off the edge of the cliff kind of way. Okay, no halfway. We're probably gonna bomb, but we're gonna test the audacious hypothesis that it is the microbiome that allows rats to decide that another type of rat is worth opening for. So what we're going to do is we're going to raise albino rats in a black cape rat environment, females. They are going to have babies. They will communicate their microbiome to the babies through a vaginal birth. And we will then test these pups that have never seen a black cape rat with black cape rats. And if that is communicated via the microbiome, then that extension of helping towards black cape rats should be transmitted transgenerationally. Yeah.
So I really love this approach to maybe get at the idea that there's cognitive states and empathy. Well, pretty tough one to study in an animal, right? That empathy is one you're identifying and working with here. There's another one, love, right? And if you were to branch this study into species like, I think it's prairie voles and mountain voles, where they mate for life and presumably fall in love, and the others that don't mate for life at all, they go with different partners all the time. You might be able to see really, interesting differences, make predictions based on levels of affection or levels of loyalty or levels of.
Yeah, I think that's a very interesting. Another way to think about this is mother's love, so to speak. And so one of my high school students actually came up with the question: are nulliparous females less helpful than multiparous females? Does the experience of having had a litter and raising that litter make them more affectively empathic towards others? And that is, in fact, what people think. The evolutionary origin of helping is supposed to be maternal care. And that's why mammals are supposed to be all that with helping. And, I suspect that that's probably not right, but that is the going idea. And one of the ways in which one of the predictions from that would be this idea that a mother who's been a mother, a female that's been a mother, should be a better helper than a female who has not been a mother. And that has never been tested by us or others. Okay, thanks a lot.
Great work, boss.
That was great.